Just give me a second. I just need to get my notes. Right. We humans have always tried to understand or select the most desirable trait in any plant or animals for thousands of years to engineer life. For example, tomatoes back then used to rot very quickly, etc. Till we found a way to make them last forever, or more, not forever, but longer. We got very good at it, but we never truly understood why we got it until we discovered DNA. DNA is basically this complex molecule that helps us understand more the genome, but we never truly understood how it actually worked. So basically, that's when what's happening.
No problem. بس حندور لكم l'écran ليكم.
So basically, the molecule structure is being used to encode information. Four nucleotides are linked together to produce code that contains instructions, right? You can change the person carrying, like, the luck of the person carrying that that code, if you can change the code, right? So we began testing a lot on it. When did we discover it? Just by trying to bombard it with nuclear radiation to generate a random plant or variation. But it was only successful occasionally. So basically, all of this kind of automation in itself. Then we wanted to pursue it further. We wanted to kind of control what DNA does in order to control the outcome of the DNA. So how that happened is by exploring bacteria. Bacteria and viruses used to always fought each other since the dawn of time. And what happened is, so bacteriophages, like these viruses, would basically go on the bacteria and implant their RNA. And what that RNA would do is would go to the genome of the bacteria and change it. Most of the time, when this happens, the bacteria dies and it just becomes the center where all the bacteriophages just replicate forever. But sometimes the bacteria actually survives this attack. And what happens is it will activate its secret weapon, or so it's called, and it's CRISPR. So technically, CRISPR, what it does is it will grab that string of RNA that was linked to the bacteria's nucleus, and give me a second. So it will go grab the link of RNA that is linked to the in DNA of the bacteria and place it in an archive. And what would happen is would look for it whenever the bacteria gets attacked again. So we wanted to kind of harvest this power to our advantage. So what if we could feed CRISPR a different strain of our DNA and tell it, for example, look for what's the gene to code for brown eyes and change it to something different. So this was the whole premise for CRISPR. And we tried to test it on animals before. So what we did was we got a series of lab rats, that got HIV. 99% of their bodies was infected with HIV, and we just tried to do this therapy on them. And by the third month, most of them just had 40% of HIV in their DNA, which was pretty amazing in itself. So with that thing, so technically we can cure diseases with CRISPR, right? So stuff like color blindness, hemophilia, Huntington's disease, etc. But it's not the only thing we can do. We can actually improve us as a species with that kind of technology, which can be a bit frightening and scary. Are you guys understanding me? Should I just continue or no? Okay. So basically, this scheme is just, what if we implement CRISPR on a human level? So, for example, we would try to help people use it on fetuses. So if we do that, the people who would be the new humans will mingle with the other humans, and that's kind of weird because if you remember, normally I don't know if you studied that, but GMOs, like for example the flavor saver tomatoes, if you put a crop of GMOs, it needs to be far from every other crop because you don't want it to contaminate organic plants because you don't know what would happen. So obviously, if we use this technology on humans, we will result to the same issues, and that's why we need to be a bit diligent. And also, the other thing with CRISPR is most diseases that we have are genome related, right? Even aging is technically a genome related cause. So why does aging happen? Is basically so you're familiar with chromosomes, etc. I suppose. So chromosomes have this thing, not thing, but more a layer of protection called telomeres at the edge of each chromosome. They're supposed to be there to protect DNA, but like, after each mitosis, that thing shrinks, and whenever it shrinks, it shrinks the DNA with it at some point, and that's where complications start happening. For example, we start having wrinkles, etc., etc. Like your hair starts becoming gray. So what if we use CRISPR to prevent that entirely, that process from happening? And we know that's already possible because some organisms like lobsters, jellyfish, Phaneron, and Nutricula are able to live indefinitely. So, in order for us to do that, it will raise a very weird ethical question: is how can you basically play God kind of thing? It's not fair, and also the humans that you're actually making would become far superior than the other humans by nature because they just have an advantage. So my counter argument there is, for example, in Europe, this is already happening. So, for example, most of Down syndrome, which is something that can be treated with CRISPR,